Hello everyone, grüezi ragazzi, bienvenue und willkommen to Cannabis Ciencia's live LinkedIn sessions. Today we have an awesome presentation for you. We have Dr. Guillermo Moreno-Sanz, who is the Medical Science Director of Khiron Heal Innovations, and he'll be talking to us about the neural pathways to Ananda. Dr. G, why don't you tell us a little bit about yourself, what you do, what your passion with respect to cannabis is, and then we're all ready to see your presentation and hear what you have to teach us.
Thank you, Christina. Thank you, everyone joining live here at Cannabis Scienza. It's a great pleasure to be here today. Thanks to Viola Brugnatelli for the kind invitation to participate in these sessions. Well, my name is Guillermo. I'm from Spain, and I'm a PhD in neuropharmacology, and my area of expertise is the pharmacology of the endocannabinoid system, which is the place or the site of action for the active compounds of cannabis, in particular THC, which is the topic that I'm going to be talking today. So basically, in this series that Viola put together, we're delving into, a little sad ways of neuropharmacology, and I wanted to bring this story really because I try to make it as open, as transversal as possible, so people from different backgrounds had the opportunity to enjoy it. That talks about how really the endocannabinoid system, we might be looking at it in parts, like what is the effect on pain, what is the effect on sleep, what is the effect on metabolism. But the reality is that we are looking at different parts of the same thing, and we might just come to realize that the endocannabinoid system is an ubiquitous regulator for many different physiological processes in the human body. So let's see if, let me start with my presentation. Here it is. So the title is Neural Pathways to Ananda. It's basically, I'm going to discuss a little bit the role of the endocannabinoid system in emotional regulation. We're going to focus largely in pain because it's not only was not only the focus of my PhD thesis and my training, but also is the main indication or the main symptom for which patients are using cannabis worldwide, the control of chronic pain. So let's start with that. And let me see. Hold
on. Do you need me to move the slides? I think last time we had the same issue. So if you could just say next slide, I think I'll be able to do that. Is that okay? All right.
Yeah, that's okay. Sounds good. So yes, please, next. So the endocannabinoid system, or ECS, I'm going to refer to just in case somebody here in the audience is not familiar with that terminology. It's a neurotransmitter and hormonal system. Basically are a series of messages that cells use to communicate among each other. And also it's a system that regulates other neurotransmitter and hormonal systems within the body. And it's the target, as we were saying, for the active molecules present in cannabis, in particular tetrahydrocannabinol or THC. Next, please. It's generally conceived that the endocannabinoid system regulates homeostasis, or the dynamic balance that keep all physiological reactions in the body in place. Maybe not all of them, but at the end of the day, we need to consider that a balance in chemical terms is not something static. It doesn't mean that we have reached a balance that does not move, But actually, as we have in the first picture, the static picture might be that of something that is immobile, but we know that this is a dynamic balance. So it's a lot of reactions happening at the same time, and the sum of all these reactions is the equilibrium or the homeostasis. Next one, please. So as a summatory of opposing motivational forces, my talk today is going to go down the route or the argument of showing that fear and love are two opposing motivational forces for human behavior, and that all feelings and all emotions are shades of fear and love and combinations of such. So this is also part of the wording that I wanted to include to, make this talk a little bit more inclusive. Next, please. So let's start with the story of fear. For if any of you do not recognize the gentleman in the picture, this is Harry Aslinger, was the founder of the Narcotic Bureau at the United States, and he just managed to eliminate cannabis from the U.S. pharmacopoeia and prohibit international trade of cannabis by fear-mongering and spreading messages of fear, such as, marijuana is the most violence-causing drug in the history of mankind. This was between 1936 and 1942. And for those of you that just wonder how is it possible that a single man in a position of power in the United States was able to create so much damage, well, consider the times that we're living today. Next, please. And then as opposing forces, we have another politician, in this case, Fiorello LaGuardia, who was the mayor of the city of New York at the time. And in 1944, Fiorello LaGuardia didn't like Aslinger very much, and he doesn't really believe all the propaganda that the Bureau of Narcotics were putting out there to prohibit trade of cannabis, calling it marijuana, to guilt minorities such as Latin America and Black population. Later on was also the communists and those protesting the war of Vietnam that were associated with this plant and the consumption of this plant that was deemed very problematic and a gateway to other drugs. So Fiorello LaGuardia didn't believe any of that. So he put together the committee of the mayor. Go find that. That's a very nice title, right? For this mayor's committee on marijuana, and put together some of the brightest minds of the time at the Medicine Academy of New York. Next, please. Including the grandmother of clinical psychology, Dr. Florence Halpern. Dr. Florence Halpern was born in 1900 and was one of the most prominent clinical psychologists at the time. And she was commissioned with describing the effects of cannabis in intellectual functioning. Now, it's funny because you've seen, I've showed you already pictures of Fiorello LaGuardia and Harry Aslinger, and I haven't been able to find a single picture of Dr. Florence Halpern. However, well, you can see that here in the intellectual functioning, this is directly from the report, from the LaGuardia report. It's public, so if you have a chance, please read it through because it's very, very interesting. Consider that this is the first time that clinical researchers had the opportunity to systematically assess the effects of cannabis in healthy volunteers, knowing the identity of the active principal ingredient of cannabis, which was tetrahydrocannabinol. So Dr. Halpern will organize teapot parties in which volunteers will consume cannabis in groups, having some tea and some cookies, and then she will be able to investigate the modifications in the intellectual functioning of those subjects under the effect of THC. Next, please. This is the obituary of Florence Halpern. It's just 2 pages. This is it's published in the American Psychology Association in PsycNet. You can read it. There's no pictures of them, but there was definitely a significant character and an extraordinary woman that took care of this of this work. Next, please. Okay, and for those of you wondering, well, but it wasn't until 1964 that Raphael Mechoulam discovered THC. How is it possible that in 1944 Florence Halper was already cognizant, or these researchers were already cognizant of the identity of THC? Well, the reality is that Roger Adams, this gentleman from the University of Chicago, identified cannabinol, cannabidiol, and also tetrahydrocannabinol in the early 1940s. What Adams was not able to determine was the position of the delta 9 double bond, which is the double bond you have in the hexagon on top, close to the CH3. So that double bond can be in position delta 9 or in position delta 8. Now, Roger Adams knew that there was a double bond, but he was not able, with the techniques at the time, to determine that position. It wasn't until 20 years later, after the development of more potent analytical tools such as magnetic and nuclear resonance, that Professor Raphael Mechoulam was able to determine the position of that delta 9. So, long story short, in the commission of La Guardia Committee was the first time that clinical researchers were able to assess the effects of THC over intellectual functioning in healthy volunteers. Next. And these are the conclusions, or these are the notes from Florence Halper. So under the effect of THC, this subject experiences some reduction in drive, less objectivity in evaluating situations, less aggression, more self-confidence. This concept of the self is the reason I'm starting with this piece of history for everybody in the audience. And a generally more favorable attitude towards himself. Please remember this, because later on we're going to see something that is going to put this in context. More favorable attitude towards himself. Next. The restraints which he normally imposes on himself. Mind you, this is 1944. The big deal is a completely different society. It was 80 years ago. Imposes on himself are loosened, and he talks more freely than he does in his undrugged state. The ability to express things which under ordinary circumstances he will not speak about are now given expression. The subject is able to retrieve information and wording that he will not normally express. Actually, we have a lot of psychiatrists telling us now that this feature of cannabis might be very interested to facilitate other behavioral and non-behavioral therapies. Next, please. So finally, as a conclusion, Florence Halper establishes that the subject holds himself rigidly to the standards of the undrugged phase and does not dream him to achieve, meaning he is satisfied with himself and is willing to accept himself as he is. Okay, this is important. We remember this piece for later on. Next. So moving forward 20 years, we have Raphael Mechoulam, which is, known for discovering delta-9 THC, but more importantly, is also known for hosting in his lab John Will Devane, who was the one, and Lumir Janus, who were the ones identifying anandamide as the first endogenous ligand of the endocannabinoid system. So a few years before, THC had been, molecular modifications of THC have been used to identify the site of action, we'll see now, which is the cannabinoid receptor 1 or type 1 cannabinoid receptor, also known CB1. And then the Mechoulam lab was able to identify anandamide, which is a derivative of arachidonic acid that is the endogenous molecule that binds to the endocannabinoid receptor type 1. Next. As we were saying, Ananda means internal bliss in Sanskrit. So that also gives an idea or reflects on the intellectual functioning described by Florence Halper. the concept of anandamide, of activating type 1 cannabinoid receptors that you have here. So both anandamide and THC are able to act on CB1 as a key analog and elicit responses inside the cell dependent upon this activation of CB1 receptors. Next, please. So what happens when we activate CB1 receptors? So this is a quote from Vincenzo Di Marzo, which is one of the most prominent researchers in endocannabinoid science. We'll actually quote him later on again. And Vincenzo Di Marzo in 1998 said that the endocannabinoid system functions are basically to relax, to eat, to help the body relax, eat, sleep, forget, and protect. And today I want to focus on forget. And when we think about cannabis and forgetting, a lot of people make jokes about, short-term memory and how the acute effects of marijuana might lead to impairments on memory, and you don't remember where you parked your car, or you cannot find your keys, things like that. But the forget we're discussing today next, it's a bit different type of forget. Christina, next, please. So this is data from Giovanni Marsicano from 2002, another, probably, one of the greatest cannabinoid researchers. And he proved, this is published in Science in 2002, that animals, that cannabinoids, anandamide in particular, it's required to activate CB1 receptors, and that animals that do not have CB1 receptors cannot appropriately extinct aversive memories. Let's see, let's work this out. So basically what Giovanni did was he took some mice and removed CB1 receptor from those mice. So these are called knockouts. So when you knock out a receptor from an animal, you have a knockout animal. So Giovanni had these knockouts for CB1 receptors. So these animals do not have CB1. THC cannot bind to CB1, anandamide cannot bind to CB1. Anything that CB1 it's there for cannot happen. So the test that Giovanni did was, okay, I'm gonna put this animal in this cage. I'm gonna let the animal smell the cage, remember the cage, get to know the cage, feel comfortable in the cage. And then I'm gonna give him a small electric shock to the feet. Animals are not gonna like that, right? So you give this shock to the animal and then the animal freezes. That's the typical response of an animal, of a mouse, when they're scared. They freeze. So predators cannot see them, because if they move, they're probably going to be spotted by a predator. So when rodents are scared, they typically freeze. Okay, so here you have two graphs. So you see that this animal, the first day, right, you put it back in the cage and the animal does not move. The second day, you put back in the cage and the animal moves a little bit. The third day, you put in the animal in the cage and moves a little bit more. And by day, it's too small to see, but I think by day 6 or 8, the animal behaves normally, meaning that the animal is able to forget that he got an electric shock in that cage, because he didn't got any other after that one. So you basically resume normal behavior because the stressor or the threat that you have has extinguished. So the animal is able to extinguish that aversive memory. What happens to an animal that does not have CB1? So you see that the animal that does not have CB1, which is marked in black, is not able to forget that electric shock, is not able to extinguish that aversive memory. and he will have the same behavior every single day, which is different from the one that the wild type or the animal that does have CB1 has. Meaning that those animals that do not have CB1 receptors and that are not able to engage in normal endocannabinoid signaling are unable to forget the aversive memory associated to that space. And this was one of the first indicators of the critical role that endocannabinoids have on extinction of aversive memories. Next. So moving forward 5 years, we had a drug in the market here in Europe, not you guys in Canada or not you guys in the US, because it was never approved by the FDA, but it was approved by the EMA. So this is a drug that was marketed for lose weight, or for weight control, and for smoking cessation. So basically, you want to quit smoking, you don't want to put on more weight because you quit smoking, you take a Complia or rimonabant. Rimonabant does the opposite of THC and anandamide. So it binds to CB1, but it's it's a reverse agonist. It's supposed to be an antagonist. An antagonist will be a blocker, a plug you put there in the receptor, and then THC and anandamide cannot bind to it. But rimonabant goes a little bit farther, and it's a reverse agonist, meaning that it has the opposite effect. So it had to be, long story short, it had to be removed from the market because it caused severe depression, anxiety, suicidal ideation, and suicidal consumption in different patients taking this drug. So it was retrieved from the market. So the clinical research led to or suggests that if you block or reverse activate the CB1 receptor, so you do the opposite of activating CB1 receptor, subjects are not able or have more difficulties remembering positive aspects of their own personality. Meaning, so you have two variables. So these are a series of patients trying to remember some words. So if the words have a positive connotation, are easier to remember than those that have a negative connotation, unless you are taking rimonabant. If you're taking rimonabant, only when the words are positive and related to yourself, self-relevant, non-self-relevant. So these are the two characteristics: positive, related to oneself. If you block CB1 receptors, these patients are not able to remember positive things about themselves. This is the exact opposite that Florence Halpern saw in 1944. Patients taking THC, not patients, but healthy volunteers, were able to accept themselves as they were. They were happy, content, not willing to drive to achieve. So here we have two sides of the coin that match exactly the pharmacology at CB1 receptors. If you activate the receptor, the subject it's happy with oneself, content, accepts oneself. If you block the receptor, the individual has more trouble remembering positive aspects about oneself. Next. What happens when you treat patients for 7 days consecutively with rimonabant? Same thing happens. We see a depression trend. We see patients that have more difficulties remembering positive things that are self-relevant, but not those that are not self-relevant. So it's so specific, the ability of CB1 to mediate this memory extinction and this memory retrieval. Next. Another clinical indication that CB1 is mediating this type of perception. This is a group from Japan that studied individuals that have a particular modulation or a particular SNP, a particular genetic variation in their CB1 receptors, so that the CB1 receptors are more active. These individuals are able to remember positive things that make them happy or more easily. So basically, these researchers divide the construct of happiness between eumonia and eudaimonia. So eumonia being the acute state of happiness, whenever you are very happy, when you're exhilarating. And then eudaimonia being the notion of your life, of you living a happy life. This requires memory, because you need to decide if your life is a happy one or not based on your memories. If your ability to make memories positive about yourself is impaired, then you will have a lower level of happiness. If your ability to make positive memories about yourself is enhanced, then your perception of your life being a happy one will be increased. And this is exactly what these researchers are seeing based on genetic modifications of CB1 receptors. Next, please. So just as a transition, here's where fear meets pain, because we're going to start with pain and we're going to go, back in time a little bit. Well, not in time, but I mean, you'll see what I mean. So basically, this is the fear avoidance model of pain, meaning that nociception is the perception of pain. So whenever your brain receives information, nociceptive information, you feel pain. Your brain gets the information that it's feeling pain, and this generates a threat. Pain, it's interpreted as a threat that might be high or low. Okay. So the decision making and the behavior that the brain needs to take now is to prioritize pain control or to prioritize, other things. So basically, if the brain prioritizes pain control, the attention, the span of the brain goes to pain control, then we can have negative effect. And the first thing that comes out in that cycle, it's fear. So fear is the immediate response to pain. So very early stages in the cycle of pain, where we have then avoidance, interference with life, negative effect. We'll see this, now we'll see what this cycle comes with, but basically remember pain, negative effect. So pain, fear, avoidance, interference, negative effect. And this is a vicious cycle, which you can break if you prioritize value of life goals. And then we can have a different approach that leads to recovery. So please keep in mind these two cycles on what we're going to see next and how the endocannabinoid system might be actually regulating this cycle. Next, please. Okay, so well, this might be, probably this is another life. When I was doing my PhD in California at the laboratory of Dr. Daniele Piamelli in Irvine, me and my lab mate, Jason Clapper, we came up luckily with a molecule that was a Phi inhibitor, meaning that it increases the levels of anandamide. So it promotes the endocannabinoid tone and the activation of CB1 receptors, but only in the periphery. We stumbled about, across this molecule that did not cross the blood-brain barrier. So we were able to increase CB1 activation outside the nervous system. And we had proof of it. I mean, we were able to. So basically what we were doing, or we were hypothesizing, is that elevated levels of anandamide, activation of CB1 receptors, either in the nervous terminal or the dorsal root ganglia, before nociceptive information, remember nociceptive reaches the central nervous system, was sufficient for animals to elicit less pain. These were all animal models. We tried in 5, 6 different models, different labs in the world were doing it. So it was not in our hands. We were very, very confident of the robustness of this data. Next, please. But what happens? It happens that in 2012, we have this small pharma company called Pfizer that had a FA inhibitor in their pipeline conducting an osteoarthritis to the knee pain study, in which their FA inhibitor was not more effective than ibuprofen, I believe, was the competitor. So this was, a cold water jar for everybody doing research on endocannabinoids. Vincenzo Di Marzo had a comment on pain, saying that, well, inhibition of an endocannabinoid breakdown for pain is not so facile or facile, right? Which is right. And one of the things that Vincenzo was saying is that maybe the model was not the most indicated, because osteoarthritis to the knee does not have enough emotional components of pain for cannabinoids to be an effective molecule in this model. And I keep thinking, what are these emotional components of pain? Because I've been working with mice and with rats, so I'm not sure that we are barking at the right tree. Because you see, we have the endocannabinoid system is present in the periphery, terminals, dorsal root ganglia, in the spinal, in the spinal cord, in the dorsal horn. You have, both CV1, CV2, and CV2 glia. And there are several neural areas that contains high levels of CV1 receptors or CV2 in glial cells that might be responsible for the nociceptive actions of cannabinoids. However, only exploiting those peripheral, and in animals, it was working great. Next, please. But then we had, these commentaries, right before I finished the PhD, saying that basically people were saying that the reason why there was such a high failure rate on translation from preclinical to clinical research, particularly in pain, is because the models that we were using for pain in animals were useless and were specifically relevant because animals do not possess a particular pathway that goes from the lamina 1 spinothalamo-cortical to the dorsal posterior insula by way of the posterior part of the ventromedial nuclei, VMPO. Please bear with me, just for 3-4 slides. You will be safer on a very different place after the 3 slides. Don't roll your eyes. Let's see what this means. Please, next. Okay, so this means that we have 2 ascending pathways and 1 inhibitory pathway or descending pathway for the pain control in humans. What Mogil and a rat is not a monkey, is not a human means is that animals and rodents and humans, we share the lateral pathway, the one in blue. That pathway gives you the intensity of pain, okay? So it's like a knob in a radio. You have the music louder or lower. But the pathway, the medial pathway, it's absent in rodents. It's exclusive for humans. And again, it goes from, those points that we've seen, the VMPO, you see that you do the amygdala, and see that it goes up to the left to the ACC. That's the anterior cingulate cortex, okay? And then we have the suppression pathway, the inhibitory pathway, which, takes care of pain suppression. We'll see that later on. But this concept of 2 pathways, the lateral pathway and the medial pathway. Next. Okay, so one starts reading because, you want to understand what's going on with this human and with this lateral pathway. And it comes out that there's psychological and neural mechanisms on the affective dimensions of pain. Affective dimensions of pain. So this means this is not how much it hurts. So it's not the intensity of pain, but the affective dimension of pain. And it seems that the ACC, you see it there, so we have the ascending pathway to the insula through the ACC. So one part goes to the somatosensory cortex, where you have all the memories stored, and the other goes to the prefrontal cortex and the amygdala, which is the way you react immediately to things, right? And so that happens that Lee in 2013 shows that this activation to the ACC, that pain causes to the ACC. So whenever you have painful nociception, right, going to the ACC, if you take THC, you block that signal at the ACC, specifically at the ACC. That's the panel to the right. Next. So, okay, if you keep going, I mean, there's not a lot of data in here, but there is some research done, and some groups that have done this very tedious research trying to understand how, the amygdala, the somatosensory cortex, and the thalamus, all these pathways of information connect. So you see that the M1 model, it's preferred in what is called the pain matrix. So the thalamus, the somatosensory 2, and the hippocampus are connected. So you see the arrows, which model is favorable. Next, please. And here you have some imaging studies showing the same thing. Administration of THC decreases the level of activation in the anterior insula, the hippocampus, and cerebellum of patients. These are volunteers undergoing experimental pain. So we don't do this kind of experiments in patients. We do it in healthy volunteers that we subject to pain. So it's not essentially chronic pain. And the CNS of the central nervous system of these volunteers is not affected by a chronic disease. But it shows that the input, the nociceptive input, produces an activation in the anterior insula, hippocampus, and cerebellum, and THC can selectively decrease that. And it also reduces the functional connectivity between the thalamus and the somatosensory cortex, and the amygdala and the primary somatosensory cortex, which were in the maps that I showed you in the slide before. Next, please. Another example. These are also healthy volunteers, 13 actually. This is not a large experiment. And we're going to see what is the combined effect of morphine and dronabinol. This is oral THC. So it's not even inhaled, it's not even cannabis, it's purified THC. So you see that the combination of morphine and THC has no additive effect on the sensory response, meaning the intensity of pain, meaning the lateral pathway. But the combination of THC and morphine has an additive effect on the affective responses of the painful stimuli. So morphine is likely acting on the descending pathway, the one in purple that we saw to the right, but cannabinoids are THC is activating the medial pathway, not the lateral pathway, to produce a reduction on the affective responses. These authors actually make the simile to the radio, meaning that the intensity is the volume. When you enter a room and there's music on, there's music in the room, the volume, if they ask you, okay, 0 to 10, where's the volume? You can give a number on the intensity. But the affective responses will be like, if you're asked how much the music bothers you, that really depends on the setting. That really depends on your likes. That really depends on you and what you find a stimulator or not. Is your favorite song, even if it is, at a 9 of volume, you might want to listen it to a 10. So that the affective component of it might have nothing to do with the volume at which the music is playing. So long story short, again, remember, THC was able to disclose an effect on the affective responses to pain, but not on the sensory, so on the intensity of pain. Next. This is a very interesting study. This is a meta-analysis of studies that test cannabis and cannabinoids in pain labs. These are all, again, healthy volunteers subject to some form of painful stimuli, taking cannabis or cannabinoids. And we see, what the authors here find is a small effect on the intensity of pain. So a reduction on the threshold of pain that is already being felt. Next. There is no effect on the threshold of pain, but there is a significant effect on the emotional components associated to experimental pain. Again, reinforcing the notion of all the imaging and all the rest of the evidence that we are seeing. Next, please. Okay, so what is the difference between pain and suffering? What are these affective components of pain? So this is not so relevant for acute pain, but it seems to be very, very relevant for, one, the transition from acute to chronic pain, and second, the maintenance of chronic pain. Again, let's go back to the cycle. Once you feel pain, you have these two ways to go. The first one being fear. So what happens with the pain onset? You have trauma, injury, strain. After 3 months of persistent pain chronifies. And then we have other symptoms such as repetitive strain, inactivity, pain tensing, poor posture, anxiety, stress, poor sleep, fail of treatment. None of that is intensity of pain. All of those are emotional components of chronic pain. Depression, disability, loss of purpose, other comorbidities. This is what generates the burden of chronic pain, not pain intensity. And we see that cannabinoids are able to tackle each one of these components of pain. Next. So, as you can see, according to these authors, pain intensity that it's mediated by the lateral pathway amounts for 0 to 3% of disability, while suffering associated to chronic pain, which is mediated by the medial pathway, accounts for 30 to 40% of disability by means of catastrophizing. Next, please. And these two pathways might be targeted with specific interventions. So you have a list of interventions. Some of them are drugs, some of them are meditation, yoga, acupuncture, other type of interventions. You can see that in the medial pathway, we have paracetamol, which we know has a cannabinoid-based action mechanism. On the lateral pathway, you have aspirin, gabapentin, other traditional painkillers, or drugs that are given to treat acute and chronic pain. And in the descending pathway, we have the opioids and other drugs. The strong painkillers that are used in chronic pain. So these pathways are also distinguishable from a pharmacological standpoint, and cannabinoids seem to be activating specifically the medial pathway. Next. And again, the pain sensation leads to several components that are affective, catastrophizing, which is the negative deficiency, stress, unpleasantness, which leads to suffering that has emotions associated to it, anger, fear, frustration, anxiety, depression, which become comorbidities of this suffering and hence of chronic pain. And all of them are mediated by the same neural pathways. Next. So what happens with the this is a little bit more of literature based on this two concepts. So first is the phenomenological study of pain relief by cannabis. This is a very, very interesting qualitative description of patients showing and disclosing what is that they feel, what is their experience when consuming cannabis. And one of the main features of cannabis in chronic pain is to improve sleep. Now, rumination and catastrophizing might happen during the day, but mainly occurs at night when people cannot sleep and start thinking about, their pain, their situation, what the future holds, how, their life is terminated by whatever happened to them, revisit trauma. So this is one of these negative cycles that largely affect the outcomes of, or the quality of life of patients with chronic pain. And again, it's all mediated by the lateral pathway. We see other chronic indications such as anxiety that are also alleviated by activation of CB1 receptors, and the same neural pathways are involved. So dorsal medial prefrontal AAC, anterior cingulate cortex to the amygdala, amplifies medicated generalized and social anxiety. So anxiety patients have the same neural pathways engaged compared to chronic patients that experience also anxiety associated to pain, to chronic pain. Next, please. And then enters Joe Cameron. Here we can take a break. Okay, sorry, maybe there was more than 2 or 3 slides, but I hope that I didn't lose everybody. It was in the title that it was about neural pathways, but we can relax a little bit now. Joe Cameron. Who's Joe Cameron? So she was 71 or 72. She was a teacher, retired teacher probably at the time, from Scotland. And he was the first individual in which scientists discover a particular deletion of a cannabinoid gene. So Joe Cameron has a defect on her Fa gene, meaning that the enzyme that degrades anandamide, that destroys anandamide, the enzyme that my molecule and Clapper's molecule wanted to block, and that was very effective analgesic in animals. It turns out that Joe Cameron has less of it, meaning that she has more anandamide. Check out those levels. She probably has twice as much anandamide as anybody else that you know. And according to the New York Times, she never felt pain or anxiety in her life, and now we know why. Check out in their hands, you can see that she has cuts and scars from hurting herself because of pain insensitivity. She was not able to feel the burn of an ironing, for example. She gave birth twice without any painkillers, and she has always an excellent mood. So here's another clinical correlate of enhanced levels of anandamide, of enhanced levels of CB1 activation, with an outcome of pain control, nociception control, and lack of anxiety and stress from a human being. Next. Okay, if science is not your thing, if you're stressed even with Joe Cameron, here's another notion, more poetical probably, of what fear and love and the balance of the two entail. Please, while reading the words of John Lennon, remember the godmother, the great mother of clinical psychology in New York in 1944, experimenting for the first time with THC and finding that activation of CB1 receptors make a person accept themselves as they are. So there are two basic instincts motivating forces, fear and love. When we're afraid, we pull back from life. When we're in love, we open to all that life that has to offer with passion, excitement, and acceptance, again, emotions. We need to learn to love ourselves first, the self. Remembering positive things about yourself, You need your CB1 receptors engaged for that. In all our glory and our imperfection, accept yourself as you are. If we cannot love ourselves, we cannot fully open to our ability to love others and our potential to create. Evolution and all hopes for a better world rest in the fearlessness and open-hearted vision of people who embrace life. Next. Okay, if science and music are not your things, first of all, I don't know how you end up in the Cannabis Scienza live recording, but okay, let's have it. If you like films, you have Master Yoda saying fear is the path to the dark side. Fear leads to anger, anger leads to hate, hate leads to suffering. You all saw those words. I gave you neural correlates for them, but if this resonates better with you, so be it. And then we have this is a picture from Alex Gray. It's not a painting from Alex Gray, which is a visionary artist from California, showing how cannabis outside the brain or outside the body and cannabinoids inside the body are able to manage this balance between love and fear, controlling stress, controlling anger, controlling suffering. Next one, please. So I just wanted to, finalizing my presentation. I think, it was probably enough for this afternoon. But I also wanted to touch base on things that we have, or I have stumbled upon in these last years. One of them was for basically, one of the psychiatrists working with me in a clinic in the UK one day said, Guillermo, we have many patients with emotionally unstable personality disorder, very young patients, 22, 23-year-olds, that are self-medicating with cannabis for EUPD, emotionally unstable personality disorder, formerly known as personality disorder. They use it mainly to control impulsivity and to reduce the risk. And well, the issue with these conditions is that, for the most part, in some cases, you can interpret them as self-medication of people that have an underlying condition, but for the most part, it's just an excuse to smoke weed. So you never know, and obviously, they're always going to be judged for the consumption of cannabis. So, funnily, or luckily enough, there is a group in Canada led by Dr. Koya, which had described in the prefrontal cortex of EUPD patients, there is an increase of FA expression. So basically, if you can see those ABCs, so A is your normal representation of the endocannabinoid system in a neuronal cleft. So basically, when a neuron is communicating to another neuron, it will send information, and then when the postsynaptic neuron has enough information, will say, stop, I have enough, and use cannabinoids for that. What happens in the UPD patients, they have very little anandamide to send this stop message. So neurons will keep firing, right? If you give exogenous THC, then you can revert the situation and bring it back to homeostasis. That's our hypothesis. And that's the reason why we believe that, considering that there is no approved treatment for EUPD, cannabis, in particular THC flowers for inhalation, should be considered in these patients. Next. A similar situation we have in PTSD, a little bit more complicated, but PTSD might probably the archetypical condition for which people self-medicate. Without a problematic, because most of these people might actually be vets, so they have other social conditions, but this one to the left is our last paper from last year, showing that control inhalation of THC by means of cannabis flowers improves quality of sleep and general mood, the ability to relate to others in PTSD UK civilians. This is not a veteran population. These are civilians. And interestingly enough, Neumeister, together with Piomelli and others in 2013, showed that CV1 expression is increased in PTSD patients. So it's the opposite situation from a pharmacological standpoint that we have at EUPD. Different regions are affected. You're going to see that the composite value, is definitely large. Maybe the largest modification, it's in the anterior circulate. So the AAC has the largest difference in CV1 expression, not so much in the amygdala. So what is the contribution of this to the etiology of PTSD? It's unclear, but at the end of the day, PTSD, it's a combination of memory and stress disorder. So this lays the ground for further investigation on the role of cannabinoids in controlling, traumatic experience, traumatic memories retrieval, and stress disorders. Next, please. And then I'm just going to give you, the final two. This is a little bit of an inside joke with Viola because, I don't have the time or the span to go in deep into these two notions, but just two of them. So what if memory is not in the brain, or not only in the brain? Because there's amazing work done by researchers at the University of from researchers from Barcelona that suggests that there is an endocannabinoid component, a peripheral endocannabinoid component. Remember that our molecule increases anandamide exclusively outside the brain. So I might be biased, I might be sensitive about modulation of cannabinoids outside the brain. But Andres Ozaita and others have very, very significant data showing how the regulation of glucocorticoids and the suprarenal glands activity mediated by cannabinoids might have an effect on memory for emotional experiences. So this is a review, kind of an old review. It has some time already, but there hasn't been any significant breakthrough on this research other than Andres' work, for all I know. I would love to see more research done on this, but consider that this might mean that stress, anxiety, even memory of emotions, when people say that you feel with your gut, right? So at the end of the day, it will make sense that this proprioception, this nociception, these messages that go into the brain, at the end of the day, they're generated in the periphery. So there has to be mechanisms in the periphery that helps us cope with this kind of stimuli, stress, fear, anxiety. So this can also be regulated outside the brain by endocannabinoids. And then probably the final one, next please, Christina, will be the mitochondria. So there is a significant mystery in cannabinoid physiology, which is the existence of CB1 receptors inside the mitochondria. Now, mitochondria are the little organuli inside of your cells that are responsible for transforming the food that you eat into energy currency that your body can use, right? So they're basically the factories of energy. The reason, and they probably have a bacterial origin. Initially, there were bacteria that ended up in a cell, and they specialize in this function. the reason why there is cannabinoid receptors, and they have their own type of cannabinoid receptors, and the genetic, the DNA necessary to make those receptors all contained within the mitochondria. And again, going back to Giovanni Marsicano and other researchers, Arnau Busquets, for example, or Edgar. This is this is not an original paper. This is a review, a letter, but this is very, very complex work that describes the link between mitochondria and memory, and how this is linked by the presence and the activity of an endocannabinoid and a cannabinoid receptor in mitochondria. So all these hypotheses that I lay before your eyes, I mean, obviously this is very tentative. I mean, it's very it's not a proven theory. I'm just jotting down and putting things together that I think make sense from the clinical experience that we see, based on the effects that we see of cannabis-based medications, of chronic pain, and particularly on the affective components of it. But there is very compelling evidence, clinical evidence, collected over 80 years that suggests that cannabinoids might have a critical role in establishing and retrieving memory that is affective, that is affects proprioception, so it's self-relevant. And we are still far from unraveling, all the mysteries that the endocannabinoid system have for us in store. But obviously, I think that this is a potential mechanism that could be easier to gather data on. So I think this is my last slide. Next, please, Christina. Yes. So this is my email again. Thank you very much for having me. It was lovely to participate in here. I hope that you guys have a good time, and that this was, both instructive and fun.
Thank you so much, Dr. G. That was a lot of food for thought, even for me. I was taking lots of notes. Just a quick question, something that popped to my mind when you were discussing mitochondria, CB1 in mitochondria, and you mentioned that the DNA encoding for the CB1 receptor on the mitochondria is stored in the mitochondria. Now, have you seen any difference in the structure or the DNA sequence between the DNA for CB1 receptor encoded in the nucleus of the cell versus the one in CB1 in the mitochondria?
So, this is way outside of my area of expertise. I would say that there's a lot of homology, because otherwise it won't be the same receptor. So there's a lot of homology, but it's nothing extraordinary that mitochondria have their own DNA. So mitochondria have their own DNA to make everything that they need, rather than making copies of it in the nucleus and then getting proteins done and back to the mitochondria. So it's normal that mitochondria have this DNA, but it's still a piece of a puzzle why CB1 receptors are present in mitochondria and what is their function. Actually, the work from Giovanni Marsicano is probably, that lab is the only one that is making work on mitochondrial CB1. Some of their students are now establishing their own labs, and such as now, for example, and following up on this lead. And hopefully we'll learn more. I would say it's likely not identical, but it has to be close enough so that still has the same functionalities. And obviously, one of the things that are really puzzling will be which is the endogenous ligand of this mitochondrial CB1 receptors, because we don't know that anandamide is created inside the mitochondria. So it's still puzzling indeed, I mean, there's like a significant part of the scientific or scientists or researchers that are skeptical about the presence of CB1 in mitochondria. I think Marcicano is the first one that tries to be skeptical, but when you prove something over and over, then you can just not deny, even if you don't understand, the significance of what you're seeing.
Yeah. All right. Yeah, thank you so much. Just want to
No problem.
remind our viewers to check out our website, cannabisciencia.com, where you can find the principles of clinical cannabinology. And then also just want to remind people to join us next Tuesday as well for a continuation in this series. And we will have a lot of other fascinating speakers. I keep seeing I host one of these sessions and I'm like, oh my God, this was mind blowing. This is my favorite. And then I go to the next session. It's like, this is my favorite. So thank you for confusing me so much now that I have too many favorites. But yeah, I'm sure that a lot of people will find this fascinating. And if there are any questions, I will go through the comments and address them. And I will email you if there are any questions that are pertinent.
Absolutely.
All right. Yeah, thank you so much.
Thank you so much, Christina.
All right. Have a good one. Bye, everyone.
Bye.